Anyone can confirm? Just type yes or no.
Yes,
Oh,
ma'am.
thank you so much. So we'll, I guess, wait for 3-4 minutes more so that because people are still joining. In the meanwhile, if people want to give their brief introduction, they can. Either you can unmute and or turn on your video and tell me, or you can just unmute and tell me, or if you're not comfortable at all, you can just text in the chat box. Basic information such as from where you are, what you are pursuing in your career, like in which degree you are enrolled in, and some basic details. Let's make this session interactive. Anyone wanna give their brief introduction? Just raise your hand. Otherwise, just type in the chat box. No one? Brief introduction, your name, which degree you are pursuing, basic details.
Hi, ma'am. Hi, everyone. My name is Koshi. So I'm from Kerala. I passed out. I have passed out my BTech degree in biotechnology and biochemical engineering from APJ Abdul Kalam University in 2022. So I took this chance to enroll in this animal physiology course for improving my knowledge about this matter.
So you are
And that's
currently enrolled in some college, or you are preparing for exams?
No, my course is completed. I have few papers left. So along with that, I'm just for improving my subject knowledge. I took this.
Currently, you are working?
And not that.
Okay. So you are planning to work and not pursue higher studies, or you want to
Yes,
pursue?
I will be not so soon. I'm preparing.
Okay. So this is what I meant when I asking that. Are you preparing for exams? Because you said you passed in from your undergrad degree in 22, right? So you might be preparing for exams or waiting for interviews, maybe.
Actually, I have few back papers left, so I'm taking this time for that.
Okay. All the best, huh?
Thank you.
No worries. Yeah. Anyone else wanna jump in?
Yes, myself, Dr. Barkha Gupta.
Yeah, hi Barkha.
Hi. I am presently working as an assistant professor in the veterinary college that is called as PGIVR, Jaipur, Rajasthan.
Okay. So
I have done my PhD in veterinary biochemistry.
Okay, nice. Why are you interested in this course?
To enhance the knowledge and for the faculty development or career advancement purpose.
Okay. So you're crediting this course, I assume,
yes,
right?
Yes.
Okay. All the best.
Thank you.
Anyone else want to jump in? Just raise your hand if you want to. I can say Sneh is there. Chandana is there, I guess. Yeah, if you both are comfortable, one of you can unmute and just tell me from where you are, what you are doing. You are here with me.
Hello ma'am, I'm Sneh. I introduced myself last meeting. I guess you remembered.
Yeah. No problem. I was about to say that Sneh is from last class, but okay. Yeah, Chandan, go ahead. I think you unmuted yourself, but Sneh said something. Yeah.
Hi, yes, ma'am.
Yeah.
My name is Chandana.
Yeah.
I'm from Chikbalapur.
Where it is?
Chikbalapur, ma'am.
Which state?
It is in Karnataka.
Okay. I'm sorry.
I'm working.
Yeah.
Yes, I'm working as an assistant professor. To enhance my knowledge, I was attending these classes, ma'am.
Oh, you are an assistant professor. What is your background
I have completed my MSc and BEd.
in
MSc in zoology.
zoology? Okay,
Yes.
okay. Then I guess this will be very easy for you. Are you following the course?
Yes, ma'am.
Ah, okay, okay. Because this is aimed for UG level.
Yes, ma'am.
Ah, okay, okay. Thank you
Thank you,
so
ma'am.
much. Yeah, yeah, no problem at all. Thank you so much for your kind introduction, guys. And as you know, my name is Ivanshi Singh. I'll be discussing in this class our week 3, like previous year questions, right? For this animal physiology course. I'm a, PMRF PhD candidate in BSB Department of IIT Kanpur. So what I want to say, as I always, I guess there are many new people here, and except Sneh, everyone is new, I guess. Yeah. And sorry if I'm forgetting someone whom I already had an introduction. So we generally start our class by asking few questions, such as all of you have gone through the week 3 lectures, right? If you have gone through all the lectures of week 3, just give a thumbs up. And even if you didn't, it's okay, don't worry. It's not a test. So I hope that most of you have gone through the week 3 lectures. You're following the lectures. I can't see a single thumbs up. Oh, you guys didn't see week 3 lecture at all. Not even like few lectures. Okay, how many of you have completed few lectures of week 3? Okay, okay, at least I've got one thumbs up. Okay, great. Achha, okay, so you guys were not able to complete the whole week. So may I know what problem you guys are facing, if anyone wants to, like, because I can see Barkha and Sneh said, like, gave a thumbs up. So Sneh, would you like to tell me that it's because of your own time management issues that you are not able to find time for completing the course, or is it something else that, like, maybe I'm just
No,
asking.
ma'am.
Yeah.
My college is
Ah,
there, so I have to do that work also.
I-
So I can't cope up both things together.
okay, okay, got it. Huh? So it's a time management issues, right? It's
Yes, ma'am.
nothing
Yes, yes.
for the duration of the lectures which we have. Okay, I just wanted to get a feedback. Uh, okay, I can see few others have joined. Yeah, so we were just discussing that. I hope that you guys have went through the week 3 lectures. If you have not, it will be my suggestion to you guys that please stay up to date with the week's lecture, because what will happen in the end, everything will pile up, right? I know that most of us are from VTech background, and we have this tendency of mugging up everything one night before the exam day. But trust me, that doesn't give you a very thorough knowledge, which we want you guys to have from these NPTEL courses. So I will suggest you, because these are what I can sense is that there are almost 5 to 6 videos, right? Till at least till week 3, there are 5 videos per week, and each ranging from 29 to 32 minutes, I guess. Right. So I'll suggest you guys to stay up to date with the week lectures, so that you'll be able to get maximum out of the live discussions. Because as you know that I'm not going to discuss any topic here, right? I'm just going to discuss certain questions and how to solve it, maybe how to solve it and all those things. And initial weeks, still things are pretty simplified and pretty easy. As you know, the course structure is always like this now, whichever course you go through, the easier and basic fundamentals are in the start, in the beginning of the course curriculum, and then more complicated stuff when we build upon those basic concepts are coming your way. So though animal physiology, we are studying it like week 1, we did suppose integumentary system. Week 2 was continuation of it. Week 3, like integumentary plus skeletal. Week 3 is also like skeletal muscle, right? So we are studying them in weeks, time snaps, but they are not like this. They all are interconnected, correct? So we cannot distinguish skeletal system from integumentary system, right? Or say cardiac system from vision receptor system. They all are working together, they are communicating with each other. So why we are studying like this? Because that is the only way to at least get some basic concepts. So if we are struggling to even follow these basic lectures, which will allow us to have some fundamental information and knowledge so that we can further in like further lectures or even in a further journey of your life, whether you are in academia or non-academia, you'll be able to build concepts out of it, right? So it is important not to mug up things, first thing. Yeah, so by this, I would like to start today's session. So whoever has watched even a little bit of the week's lecture, can you brief me what we have discussed? Just raise your hand. And even if you know only one word from this week's session, it's completely fine. Okay, just raise your hand. We'll talk. Yes, Mansi, go ahead.
So, ma'am, week 3 is basically dedicated to muscles.
Yeah, you were not audible earlier. What you
Okay,
said?
ma'am. Ma'am, I'm saying that this week is fully dedicated towards muscles, like the from the very basic to the very complex mechanisms, how the muscles helps in contraction and relaxation. So to have an overview, first of all, we are learning about muscles. Muscles is there are three types of muscles which is based on the force they generated. These three types of muscles are skeletal, smooth, and cardiac. As the name suggests, skeletal muscles is present in the skeletal system, which helps us in voluntary functions. Smooth muscles are present in our alimentary canal, which helps in peristalsis. And the third type of muscles, cardiac muscles, which is present in the heart. And then we study about the arrangement of the muscles. Then we have learned about the arrangement of actin and myosin filaments, plus the electrical activity they generated to help the muscle to move. And then we learned about excitable and non-excitable tissues in the muscles. Then we learned about the structure of the muscle fiber, like sarcolemma, A band, I band, H line, um, and the dark region, light region. Then we have learned why we are learning the function of actin and myosin, because in the upcoming 5 to 10 years, biology will be working upon 3D printing of organs. So the structure of each and every portion of muscles is compulsory. And that in the last lecture, sir taught about as the structure of actin, which has two heavy meromycin chains and four light meromycin chains, um, and actin, which has three components, which is actin, tropomyosin, and troponin. And yeah, this is the whole overview.
Very detailed summary. Thank you so much, Mansi. Yeah,
Thank
so
you,
it
ma'am.
was the basic overview, as Mansi mentioned, that we went from molecular players to organ system arrangement scale of studying animal physiology. So if you guys have missed the lectures, I request you all to please watch the lectures, right? Because then only we can have discussion. These are discussion sessions. These are not lecture sessions. So try to keep up with the lectures. Now, Mansi mentioned one point that we generate electricity, right? In our muscles, the electricity is there. Even if you haven't gone through the lecture, it's a very basic thing, right? Because all of you have at least have completed your undergrad or at least in the undergrad, right? So we are generating electricity. So I have one question which I want to put in front of you guys so that you can think and help me out in answering this question. See, the basic property of electricity is what? Whenever you see electricity, what is the first thing that comes to your mind? Charges, right? We have charge distribution. In our body, what are the charged entities? Can anyone name some or at least guess if you want to? What are the charged entities in our body? What provide charge to our system? Yes, Sneh, please go ahead.
Ions.
Yeah, correct. Ions. So if you want to build upon it, do we have only one type of ion, like negative cations or anions, something like this? Do you have any idea about it? Anyone of you? We have a distribution of ions, or we have particular set of ions?
We have both type of ions, cations and anions. Maybe electrolytes also.
Yeah. So from the next time onwards, kindly raise your hand, because so that we can organize the session. Huh? Thank you so much for the answer. So yes, and why we have that? If we are charged species, right? We are saying that we are generating electricity. Electricity means predominance of which charge, negative or positive? Hands up. Persistence of electricity means what? The axis of negative charged ions or positive charged ions? Wild guess. Anyone? Okay, someone has texted me. Okay. Yes, Mansi, go ahead.
Ma'am, both types of charges are present.
Both are there, I agree. But we are saying that if both charges are present in equal quantity, then we will be electrically neutral species, right?
Yes,
We
ma'am.
won't be charged. So I am asking, we produce electricity is present in our system. So what it indicates, that what kind of charges will be in
There are
excess?
difference between the flow of charges. For example, if we see the neural activity.
Can you speak closer to the mic, because your
Yes,
voice
ma'am.
is a bit.
Ma'am, there is a difference between charges in the extracellular and the intracellular level, because when we see the neural activity, there is a difference between sodium and potassium ion. So,
Yeah. So by this, what I mean to say, you are correct that we have different distribution of ions. This difference in the distribution of ions is what provide us the electricity. What electricity is? It is the potential difference, right? Potential difference of charges, if we are talking about biological system, it will be concentration of ions or solutes or other things which are present in a cytoplasm or extracellular matrix, right? So presence of electricity assures us that there is a distribution of ions. Now, if you guys are aware of neurons, neuronal activity and neuronal excitation and all, so from the basic electrophysiological studies, we are aware and we know this, that outside of the cell, there is access of sodium ions. Inside of the cell, there is access of negative ions, right? And also, if you don't know this, and if you guys remember, there is a electrostatic force generation when there is ATP formation of photosynthesis. You guys remember that? H ions are pushed out to the periplasmic space of cells so that they can re-enter via ATPase molecule, which is a membrane transporter. When H ions pass through the ATPase transporter or ATPase, it converts the flowing energy into ATP, right? It's a mitochondrial process, ETS cycle at the end of the day. So we need this difference. This difference is the one which is providing us energy. How we can say this? Because when we are generating the electromotive force, which is due to the differential distribution of ions, is leading to the generation of ATP. So you guys are here with me till now? Give a thumbs up if you guys are able to understand and follow what I'm saying. Okay, I have less than 50%. Okay, I'll try to simplify it. What I'm saying, we are saying that the thing is, if you guys haven't went through the lectures, in the lectures it was discussed that muscles are the place where there is some contraction and electrical activities associated with it. So even if you have not gone through the lectures, you guys have experienced this at least in winters, for example, if you're wearing like woolly clothes or shoes or those things, right? So if you're sitting for quite a while, then you walk and then you sit again and you're touching any other surface, there's a little bit of shock to you, right? So that shock is because of what? Because your body has accumulated charge, which was not able to travel through the ground because you were wearing some heavy boots. So it was accumulated in your body, and the moment you're touching an open surface, the circuit gets complete and you get a little bit of shock, right? This is what I guess most of you have experienced. Now, what I was indicating towards that we are generating electricity, it means that there is differential distribution of charge. Why we need differential distribution of charge? Because this difference in the distribution of charged ions is the major cause of generating ATP. If you guys don't know how ATPs work, it is ETS cycle, everyone aware of? Electron transport chain. Give a thumbs up if you guys have even heard of electron transport chain. Just heard of. I don't want you guys to know all the things about the cycle. But even if you have just heard about it, you have basic idea that, okay, it is a cycle. Electron transport chain. Okay. Very few. Okay, so Kajal, I guess you are new. If you're comfortable, you can unmute, and can you give me a very brief about ETS? I don't want the component name. I don't want anything like bookish. Can you just explain me the basic thing what ETS is? If you're comfortable, you can unmute. Otherwise, you can just type in the chat box. And if you're typing in the chat box, inform me that you're typing in the chat box. Yeah. Okay, cool. So meanwhile, when Kajal is gonna tell us what ETS is, so electron transport chain is a very, very basic and conserved process from very early beginning of life. Because for survival, we need energy, right? Everyone agrees with me till here? Just give a thumbs up. For survival, every single living organism needs energy. Give a thumbs up if you agree with this statement. Yeah. So for energy production in our body, the energy currency is ATP, right? How ATP is generated? The basic principle through which this energy molecule, like I'm able to talk, I'm able to move my hand, everything is an active process, correct? Because I'm doing it actively. Anything which we do actively, energy is required. That energy comes from mitochondria, which is the powerhouse of the cell. In mitochondria, how that, because we are calling that organelle as a powerhouse of the cell, but how that powerhouse of the cell is generating ATP, that should we know, right? Then only we can say, okay, this particular organelle or this particular area in a cell is responsible for the generation of ATP, that this is the source of ATP, this is the source of energy, right? So in mitochondria, there are membranes, obviously, because we are eukaryotic cell. Eukaryotic cells have what? We have membranes and membrane bound organelles. So one of the membrane bound organelle is mitochondria. In mitochondria, in membrane, there is a very, very smooth distribution and flow of electrons, flow of electricity, right? This was the basic definition of electricity. What is electricity? Is the flow of electrons. The flow of electron occurs because there is a potential difference, right? So a very beautiful chain of certain proteins are accumulated in the membrane of mitochondria, which helps in generating ATP. How they generate ATP? They generate ATP by transferring their electronic energy. When the electrons are flowing, they're transferring the energy in such a way that positive ions are accumulated outside the cell. Those still, it's a periplasmic space that I don't want to use difficult terms because apparently most of you guys have not heard about ATS chain. So what will happen? More of positive ions are outside, less of positive ions are inside. Now I'm providing one opening for the positive ions to go through, because why they want to go through? there is more number of positive ions outside, right? There will be more concentration energy associated with these molecules. They will be figuring out how to like leave this place so that they can be free, because there are a lot of them at one place, right? Collisions will be there. So for that, the moment they see an opening, they will rush through the opening, correct? It's just like just take any example of say a pipe through which water is flowing, right? So if the water is at a very high speed, that you have turned on your tap completely, the maximum, but you have a thumb on the opening of the pipe, you feel the pressure, right? You feel the pressure of the water which is coming from the from your tank. But the moment if you even lose your thumb a little bit and you allow it to open for a little while, or even a little opening is allowed, you see huge water rushes through it, right? Why this is happening? Because there are water molecules inside which are facing pressure from the tap, because we have turned it on completely. They're feeling the pressure and they have no space to go, right? So the moment they receive even a small opening, they rush through it, correct? So this is the same principle which is found in organisms also. So if there's a lot of positive ions outside, this is the membrane, lot of positive ions outside, they want to escape this place because this is not a comfortable situation for these ions. They are bumping into each other, they are moving here and there. So the moment they will see any opening, they will rush through it with high energy, right? And this energy, so this opening, what I am saying, this opening is what provided by a protein which is called ATPases. Okay, so ATPases has an opening. So the moment more H ions are accumulated here, they will rush through the ATPase with high energy, and ATPase has certain mechanism associated with it. What it will do, it will convert this energy which was passing from high speed and high concentrated hydrogen ions into ATP molecules, right? Again, here the thermodynamic property is, which thermodynamic property is being used? Can anyone wanna guess? Kajal and Mansi, can you put your hands down, because your hands are up since, yeah. So about this question, when I'm saying that the high flow of the high energy of the flow of hydrogen ions is getting converted into the ATP molecules. So what we are suggesting, what I want to say via this, there's a very basic and very universal thermodynamic property which is conserved here. Anyone want to take a guess?
Exothermic, ma'am.
Sorry?
Exothermic.
Barkha, exothermic what? Exothermic means
Release of energy.
Release of energy is one thing, but the basic principle which is conserved here is energy conversion. Energy can neither be created nor be destroyed. It can only be converted from one form to another form. Correct? Exothermic, endothermic, these are the parts of how energy is released or used. Correct? So the basic principle is the concentration energy of hydrogen ions is converted into chemical energy in the form of ATP molecules, right? Because in the ATP molecules, we have added phosphates, which are high energy bond, high energy chemical bonds. So we have converted concentration energy into chemical energy. So it's an energy conversion. Correct? Everyone here till, even if you're not able to get the whole concept, but are you guys able to understand the basic things which I'm trying to make you guys understand and imagine and feel? Okay, yeah. So, okay, we started with something else and we ended up somewhere else because you guys were not aware of ETS. Okay, Kajal has texted us about the ETS. So she says, collection of proteins bound, she's explaining ETS cycle. Okay, guys. So collection of proteins bound to the inner mitochondrial membrane and organic molecules such as electrons pass through in a series of redox reactions and release energy. Okay. Okay, no worries, no worries. If you don't know about exact terms or those things, you should not worry about it. I'm only concerned about your concepts. Your concept should be cleared. It's okay, no problem. Yeah. See, ETS means electron transport system. ETC means electron transport chain. So they both are fine. Mostly people go with ETS. Okay. ETC, sorry. Mostly people go with ETC, electron transport chain. S stands for system. So electron transport system. Yeah. So while we were talking about this distribution of ions, electricity, and all. So the question which I wanted to pose you, these were not even the question which I wanted to ask you guys. The question which I wanted to pose you guys, why don't we get electrical shock? We have electricity, right? Everything around us is also charged. Then why we are not getting electrically shocked 24/7?
Because it might be in microampere.
Sorry?
The current which are flowing in our body might be in microampere or smaller than that, so we can't feel the current.
It is not exactly that, because I don't know the exact number, but if you calculate the electricity or the power generated by the mitochondria in watts, it is more than what we get from a literal lightning, like thundering lightning, right? The power, the energy associated with the thundering, more than that is associated with the mitochondria itself. So if you have this amount of energy, this amount of electrical energy, why we are not getting shocks or other things which are associated with it? Why we are not dehydrated? Because wherever there is electricity, there is heat generation, right? And heat leads to evaporation of water, leading to dehydration. So why we are not dehydrated always? Anyone wanna take a wild guess? Okay, someone has texted me. Due to high resistance. High resistance will again suggest that we have high temperature, and we have a constant temperature of 37 degrees Celsius. 37 degrees Celsius is not a high temperature. Yeah. Keep this question in mind, and we'll discuss today's, like for this week's question, and we'll try to answer it maybe during this session or maybe in the next session, because I have one more question by the end of this session which I want to discuss with you guys, and that question might help you in understanding two very fundamental biological and physical properties. Okay, so I hope so. So let's start with our today's discussion. These questions are pretty easy, but still I'm gonna give you guys 1 minute each for every question so that you can read it properly, understand the question, give me answer in the chat box, and also you should know, if you know the correct answer, it's really good for you. But as I always say, you should also know why the other answers are wrong. Done? So this is our first question for today. So our question states, the heart is majorly made up of, is it smooth muscles, cardiac muscles, skeletal muscles, or none of the above? So you guys have 1 minute, just drop in your answers, and I want all of you to participate, at least in answering, right? Okay, I've received some answers. I guess all of you have answered it, right? It's pretty easy. It's cardiac muscle at the end of the day. Now, can anyone tell me what is the basic difference between smooth muscle and cardiac muscle? Yes, Mansi, go ahead.
Ma'am, in cardiac muscle, there is a special connection building interconnected disc which helps in the connections of one cardiac muscles to the other cardiac muscles, while this connection is missing in the skeletal muscle.
I asked about smooth muscle.
Yeah,
Difference between smooth
yeah.
and cardiac.
And second difference is the presence of striations. Striations are present in the cardiac while it is absent in the smooth muscle.
Great. So I don't think so that we have to dig into it. But as many of you might know, that I want you guys to look the real things and not the representative things. You can clearly see that this is the representation how cardiac muscles have been represented in multiple books, papers, and those things, right? But this is the real image of how cardiac muscle look under microscope, right? So they're pretty well oriented, well organized, and we can clearly see the striations, the junctions, right? So before going to, again, it is just a suggestion for you guys, before going for any representative images, try to look for the real images. They will give you better learning. At the end of the day, you need to start imagining things in the real time perspective, not with respect to the representative way just make us understand in easier way, but at the end of the day we need to deal with the real system, right? So without discussing much on this, I guess most of you guys are well aware of smooth muscles, cardiac muscles, skeletal muscles, the difference between them, the similarities between them. Okay, so we'll move on to the next question. So what is our next question? And also remember, in these slides, I'm providing you guys some different links. These are not the links of any particular book or something like this. These are the links for proper journal articles, review articles, which will help you guys in understanding the uh, vastness of the topic, because whatever we are studying here, it is just a very basic introduction and fundamental things for you guys. But I want you to know that whatever you are studying right now, there has already been multiple things which have been built up upon it. So you guys should know the extent of the knowledge distribution which is available for particular things. So I try to provide good and easy to read review articles most of the time, so that it will be easier for you guys to even understand and also to follow. Another suggestion, if you guys are not able to follow and you find it difficult to read journal papers or research papers, don't worry. Gradually, and like if you keep reading, gradually you will understand it. So don't demotivate yourself that you are not able to understand what this paragraph meant, because sometimes scientific language is difficult to understand until unless you are directly associated with that field. So don't worry about it. Reason, always go for review articles first if you want to learn something. Great. Done. So our second question states, digestive tract is mostly made up of, first, smooth muscles, cardiac muscles, skeletal muscles, or none of the above. Your 1 minute starts now. These questions are very, very basic if you guys have went through the lecture continuously. So it's kind of easy to go lecture. We are not yet in we are not in the uncharted area till. Okay, I have received few answers. Okay. 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 Cool. Okay. Yeah, obviously the answer is smooth muscles. Can anyone tell me one property of smooth muscle? Mansi, can you put your hands down? I guess they are up from the past question. Yeah. Okay. Anyone else want to try? I'll come back to you, Mansi, but I want others also to speak. Even if you have not gone through the lectures, from the name, what you understand. Don't worry, we are all starting from a ground zero level. Don't think that if you haven't gone through the lectures, you won't understand. I'm here for it. It is my duty to make you guys understand, right? So what you understand from the, because all of you have answered it. which means one way or another way, you have heard about it or know something about it. So I just want to know that something what you know about it, right? So those who are quiet in the class, you guys are comfortable, can you unmute or just raise your hand if you want to give answer and type in the chat box if you don't want to unmute. But I want people who have not spoken yet to be active. See, this is a group learning. This is a discussion session. We are not being judged here. We are all here to learn. So even if you know a little bit about something, it will be, you might not know that that particular information might help someone else to connect their dots. Okay, so don't underestimate yourself. Okay, so we are getting few one-word answers about smooth muscles, elasticity, movement, gastrointestinal motility. Okay, we have a particular word for this motility, right? Peristalsis. You guys aware of it? If you guys are aware of peristalsis, give a thumbs up. Okay. So other guys, you guys have not heard about peristalsis? Oh, okay, I am receiving messages also. I have to keep track of who is messaging and who is like giving thumbs up. And what if you guys both, like all of you are saying, my counting goes bad. Okay, no problem. Yeah, so smooth. Barkha, would you like to give it a try? What are smooth muscles and peristalsis, if you're comfortable?
This is the movement in internal organs, ma'am.
Internal organs or particular organ?
Basically,
Which
internal organs.
Acha. But peristalsis is not present in our muscles, right? Muscles are also organs, I assume.
In hollow organ, we can say
Digestive tract.
digestive tract.
But in digestive tract. Why we need such motion? If you wanna give it a try.
Because of the
Go.
for the movement of digested material.
Or can we say that, okay, let's do a thought it's okay, I'm not asking directly to you, I'm asking to everyone else. Let's do a thought experiment here. If you guys are not aware of peristalsis, currently I don't have anything for you guys to demonstrate that. But okay, let's take this. It's a, okay, you guys are able to see, right? Let's take this, and this might not help you understand like more clearly, but I'll try to explain it because I don't have anything nearby me. So if this is your digestive tract, the moment you eat food here, right, you chew it, you broke down it into smaller particles. Now, when you were chewing it, it was the energy and power of your jaw, right, which was doing the work. But you don't have power in your neck region or below that region, right? As you move from mouth to stomach to intestine, right? You don't have jaws like thing in your stomach to do the work for you, or you do the work voluntarily, knowingly that now food is there in your stomach, now you have to chew your stomach, right? It's nothing, something like this is not at all happening. So if this is the tube of your esophagus, let's say this is the food pipe, this is your food pipe. So till here you can control your motion, right? By chewing it, by changing the rate of chewing, by changing the frequency you are chewing, by even changing the duration of chewing, right? So it is still in your control. You can control this aspect of eating food. At the end of the day, why we eat food? We eat food for the energy, right? So energy is generated by the distribution of ions, correct? So everything is related. This is what I meant when I was saying that though we are studying different things on different week sets, everything is related. So if you're eating food, till here you can control, till your mouth is there because your jaws are working voluntarily. But the moment you engulf your food and it passes the food pipe, there is nothing else for it. You are not pushing your outers like this now to let the food go through it. So there, these muscles are associated. These muscles are called smooth muscles, right? So these smooth muscles, what they do is they continuously do this. For example, do this, right? Like this. So if one muscle is doing this, whatever the food was here, it will pass down to the next part. Now the second muscle was here. It will again do the same thing. The food particle is now here. Now the third muscle was here. Now it will pass down to this. So this motion of muscles, letting the food go from the esophageal opening to till the end, is called the peristalsis movement. When you vomit, vomit indicates what? That it is in the opposite direction of the ingestion, correct? So in vomiting, there is reverse peristalsis. When the peristalsis was happening, this, that the first muscle group, second muscle group, third muscle group, fourth muscle group, this was the ideal way. But due to certain reasons, what is now happening, that the pattern has reversed. This is happening. So what is happening? All the food now starts to coming from bottom up, and it leads to vomiting at the end of the day. So this peristalsis movement, or the movement provided by the smooth muscle, is very, very important for the digestive tract, because this is the sole process through which transportation is being done. Blood is not associated with the digestion system. Blood is not carrying any food particle or any food. It is carrying the digested and assimilated molecules. I hope that this is clear to you guys. Just give a thumbs up if you are following till here what I said, that blood is a circulatory system, but it is not circulating food. It is circulating the molecules which have been assimilated and digested by the digestive system. So in the digestive system, how the transportation is happening, it is not gravity only, right? That we eat and with the help of gravity, everything is happening. No, nothing like this. It's a very controlled and regulated way. Why we need it in a controlled and regulated way? Because every food is associated with different types of enzymes for it to be digested. Correct? Everyone agrees with me? If I eat protein, there are different set of enzymes which will digest protein. If I eat fat, there are different sets of enzymes which will emulsify it. If I eat carbs, there are different set of enzymes which will digest carbs. Correct? So if the enzymes are different, the substrates are different, their activity will also be different. And if their activity is different, which means they require different time to do their work. Everyone able to connect? Give a thumbs up if you're able to connect what I'm saying. I want you guys to follow. Okay. So those of you who are not able to follow, can you guys drop your queries in the chat box if you are not comfortable in unmuting and telling me? Because I want everyone to understand certain very, very basic things. These courses and these building up, knowing things. For example, you know, smooth muscle is the one which is like covering all our digestive tract. But what is the significance of smooth muscle? Why only smooth muscles are there? Why not other muscles? We can ask this question, right? If all the muscles during the beginning had equal probability of being any muscle which they wanted and wherever they wanted to go, then why only we are seeing smooth muscles in the digestive tract or soft organs? Why we are not seeing cardiac muscle in the digestive tract, or why we are not seeing skeletal muscle in heart? There has to be reasons, right? It is not like that we are studying, okay, we observed in heart, there's a different type of muscle, which is cardiac muscle. In intestine, we have different type of muscle, we name it smooth muscle. No, there are reasons associated. This is all research is about, right? Why something is there? What is the role and function of it there? So there is a dedicated function of smooth muscle, which is associated with the digestive tract. That is peristalsis, elasticity, transportation, which was not as par as if skeletal muscles were there. Why? Because skeletal muscles are the ones which generate more force. And if we are generating more force, imagine your internals. For example, I don't know how many of you guys have experienced this, but when you don't have anything for quite long, you are hungry, your stomach starts to make sound, right? I guess everyone can relate to it. That the stomach. So correct, everyone has experienced it, right? If you don't eat food for a while, your stomach starts to make weird sound. What that sound is, the sound is your stomach is also an organ which is lined by muscles. Those muscles are contracting. They're contracting, but as there is nothing inside, so they are just contracting in empty space. And in empty space, there are liquid in the stomach, and due to which that sound is coming. Correct. So if you have food in it, when the muscles are contracting, the food will be getting churned. Correct. And it will be retained in the stomach depending upon what you have eaten, depending on what enzymes are now currently acting on in the stomach, so that it can be digested and assimilated in the intestine. Correct. So smooth muscles are associated with digestive tract because they have this property. They have this property. That is why they are associated with the digestive tract, and they are not associated in the heart. Because in heart, we need constant relaxation and contraction. In skeletal, it depends upon our voluntary action. If smooth muscles, for example, are present in place of skeletal muscle, what do you guys think will happen? Just a brief guess. What my question is, skeletal muscle, for example, I have this skeletal muscle right here. So instead of this, what if I have a smooth muscle? What will happen? Yes, Sneh, go ahead.
I guess we can't lift things.
You cannot lift things.
Because I think smooth muscles are weaker than the skeletal muscles. So we can't hold up things for a while.
So our tension won't be that much. For example, I can hold this cup for quite a while if it is not heavy for me. But even this cup is not heavy. But maybe after 1 minute or 2 minutes, I will start feeling tired. Correct? I will start feeling tired. But if the smooth muscle is there, I won't be even able to lift it up. And even if I was able to lift it up, as soon as I lift it up, I need to drop it. Why? Because my smooth muscle don't have this ability to have such high force or have such high persistence or have such high tension in it, so that they are able to retain my hand's position. Correct? So internally, if we have skeletal muscle, say for example, but we don't need such force. We don't need high force to churn food or to not churn food only, but to transport food. So then why will evolution or the organism will adapt and have skeletal muscle in place of smooth muscles? Smooth muscle is playing very significant role. Skeletal muscle is playing very different role. Cardiac muscle is completely different group, right? But they are different. Why? Because they are performing different functions. If you switch them out, you will still be able to perform the function, but it won't be at its maximum. If I have a smooth muscle, my job was to lift it up. I was successful in it. If instead of skeletal muscle, I have smooth muscle, I'm successful in it. But my further action, my goal was to keep it still. That won't be fulfilled by the smooth muscle in place of skeletal muscle. Correct? So we can interchange muscle, but their function determines the optimum level our system is tuned to. Correct? Okay, so without discussing much, and again, as you can see here, that this is the real image of smooth muscle, how they look under microscope. Correct? Do follow these links, guys, if you are even little bit of free. I know it's too much to ask for because you guys need to do your work. You guys need to follow the lectures. Just give it a try. If possible, good. If not, then obviously you guys have these slides. You can always come back to it. So our next question is, which of the following is the basic functional unit of contractile muscle? Myoblast, myocyte, myotubes, or myofibers? Your time, your 1 minute starts now. Okay, I'm getting answers. Try to read the question again, guys. Because here comes the structural part of it. Remember what was muscle, what was inside the muscle, and what was inside that muscle. We are asking about that inside. There were different structural levels for muscle, right? So which of the following is the basic functional unit of contractile muscle? Basic unit I'm asking. And okay, let's see if we can eliminate certain options. Okay, if we don't know the correct answer, then we can try to eliminate certain answers, right? So here the question is asking about which of the following is the basic functional unit of contractile muscle? We are asking about the organs functional unit or tissues functional unit, right? If we are asking about functional unit of a tissue or organ or those things, we won't say cell's name, correct? Because cell is the functional and fundamental unit of life. But we have moved on. We are not asking about life. We are asking about another level, which is the organ or tissue level. So for organ or tissue level, we can, for this particular question, we can eliminate which option? Myoblast and myocytes, because these two are cells, correct? So we can remove these two options at least. So now what we have left with, we are left with myotubes and myofibers, correct? So even if you are not 100% sure about what are myotubes or myofibers, but if you can, for example, have any basic knowledge about, say here, if I'm talking about, right, hair. So in hair, everyone, I hope that because due to social media, you guys might have seen many electronic images and many microscopic images. So when we see hair in the microscope, we see it's a thread, because for us, it's a thread-like thing, right? So it's a fiber-like thing, correct? It's a fiber. Now, when we look inside, there are different areas in the hair. There are different layers in the hair, which indicates that there's a pipe and pipe kind of orientation, indicating that what we are seeing here, that this is the macro organization of things. There's still something which are inside, correct? So fiber means there are different units which came together to make a fiber. Tubes are the smallest things. We call this fiber. If something is there, okay, this is cotton fiber, right? That this is cotton's fiber. So that cotton fiber is made up of different sets of proteins inside that fiber. Then only it is a fiber-like structure, right? Because there is not a single molecule which is extending from A point to B point. You cannot have that. There are multiple small molecules which are joined together in a very strategic manner to create a one clear-cut fiber. So from this, even if you guys are not aware of the correct answer, so we have completely eliminated the first two options. We had now doubt in last two options. So from basic, the things which we observe in our surrounding, we can see, okay, fibers are the macro things. Hence, it cannot be a it cannot be a functional unit. Functional unit because suggests that it has to be the fundamental and the only smallest unit possible for that particular thing which we are asking about. So we can answer about we can go about this question like this, okay, without having much prior information. I'm not saying that you guys don't have any, should not have any basic prior information. Obviously, having some information help us in solving such easy questions when we are not 100% sure about things. Okay. So the correct answer is myotubes. And those of you who have answered myofibers, I would suggest you guys to go back to the lectures, because sir has explained it in a really, really good manner. He has, again, as I say, he builds everything from the ground zero to ground infinite. Okay. So this is a very colorful image. Okay, how many of you have seen such stained or confocal? These are not confocal, but are microscopic. Let's say, how many of you have seen such colored and vibrant images? Just give a thumbs up if you have seen. Even on social media, I'm not talking about description of those things, how to do that, nothing at all. How many of you have seen it? Okay, very few. Chalo, okay, no worries. But these are tube doesn't always mean hollow. Generally we say that tubes are hollow. So if you go through the lectures, you will understand why we are calling it tube. We are calling it tube because this is, for example, this is one cell, right? So earlier it was like this. This was the whole cell, if you can see. This was the whole cell. But what myotubes are multiple cells which have came together. See these. Now you can see it's an elongated tube-like structure. So this is not one single cell. There were multiple cells earlier, but later their junctions were removed and they conjoined. So now they are creating one single tube-like structure, but still they are different cells inside, because we can see different nucleus inside it, right? So that is why we are saying it as a tube, because generally cells are like spherical, round, and like this. They are not in tubular shape. So when we join multiple cells, especially for the muscle tissue, multiple cells come together, then they join together and their interjunctions are dissolved and they create this tube-like thing. And in biology, chances are you might not have hollow things, right? There will always be something present. Even cells are not hollow, they're spherical, they have cytoplasm. ECM has extracellular matrix and different things are there, right? So chances are they might not be hollow, because hollow means there is, then the concept of pressure will come, positive pressure, negative pressure. We face, like, if you guys have traveled via plane, you feel the air pressure in your ear, right? Whenever you're taking off or when you are landing, it's because of the pressure difference. And why that pressure difference? Because our system is maintaining a consistent negative pressure inside us, and the pressure from outside is like higher for us, so that we are able to maintain our system in a like a closed system. But the moment we change the pressure, things starts to go haywire, right? So most of the time hollow things won't be present in mammals specifically. In biological system there are exceptions, and I cannot say for sure, but with respect to mammals, having hollow things is quite a difficult thing to imagine, at least for me. And I don't have any right now, I don't have any proof for it, but I might get you in the next lecture maybe if I remember. Okay. So this diagram, by this diagram, what I want you guys to see, this was some experiment from a paper. I've given the link here. You guys can go back. You might not be able to understand, but you can just read the description of the image, what it says. So everyone is aware of stem cells. We have discussed in the last class. And even if you guys are new, I hope that you guys are aware of stem cells. If yes, then just give a thumbs up. If you guys know what stem cells are or just heard of it, what stem cells are. Okay, I have reactions with me. Okay, great. So I assume that you guys know what stem cells are. So these were the experimental setup in which the stem cells were, this is first image is the stem cell. Then they have given some stimulus to the cell so that they start dividing into one particular type of cell, which is the myoblast or myocytes, right? Which are the muscle forming cells. So now after few days after the stimulus, you can see how their appearance has changed. You guys are able to observe it. Yeah. Can you see literally the physical difference in the orientation of the cells? These both are the same cells, but the moment we give stimulus for these cells to get differentiated or get converted into muscle cells, see how they are progressing. Okay, they're completely changing their structures, correct? And this is the colored image of the same thing. And in the bottom row, these are like actin, myosin, nucleus things. Just don't focus on these first few panels, just focus on the last panel. So what last panel is saying, this is how your muscle look under microscope if we are doing in vitro experiments. So from this cell to this organization, a muscle was formed. Okay, this is how we do experiments in our labs. If we want to understand some contractile property of a muscle or some other property of a muscle, or just to test how different muscles behave in different environmental conditions. So we do such in vitro experiments before going to animal studies. Okay. Yes. So this was for your basic information. We'll move to our next question. Our next question is, the muscle condition that is associated with loss of muscle mass and force is called muscle strength, sarcopenia, fatigue, or tetanus. I'll give you guys 1 minute for this question. And also try to form things that why other options are wrong or what the other options mean. I'll repeat the question. The muscle condition that is associated with loss of muscle mass and force is called muscle strength, sarcopenia, fatigue, or tetanus. Okay, I have received few answers, but very few. I still want other people to answer. Even if you are wrong, don't worry about it. Just even your wildest guess, if you think, okay, this is what I'm feeling, this option might be the right answer, just drop in the chat box. Okay, we have got few answers. Let's see what is the correct answer. The correct answer is sarcopenia. So kudos to all those who were able to get it right. And don't worry if you were not able to get the right answer. You were not able to get the right answer. Don't worry about it. So anyone wanna tell me about fatigue? What is fatigue? Anyone? Everyone has heard someone saying that they are feeling fatigue, right? That they are tired, fatigue. So yes, Koshi, go ahead.
Ma'am, fatigue has nothing to do with functionality of a muscle or its strength. Fatigue is more like tiredness and low energy, such a situation.
Not necessary. Fatigue is actually associated with muscles. So we say muscle fatigue, if you remember. Like, have you heard someone saying muscle fatigue? So what muscle fatigue is, for example, in the earlier example, what I said, if I want to hold this jar for too long, for 1 minute, for 2 minutes, fine. But the moment I'm holding it for extended period of time, say half hour or 1 hour, I'll start feeling tension here, right? In my arms. So I would like to drop this and just take rest. So what I'm feeling is the fatigueness in my muscle. That is muscle fatigue. Why we are feeling fatigue? Again, it brings back us to what? It brings back us to the basic functionality of muscle, which was to relax and contract, right? Relax and contract. Relaxation and contraction is a energy consuming process. Energy consuming process means ATP is being used for this function. When ATP is being used, for ATP to be used, it will be used for metabolism, because we eat food. From food, we take glucose. From glucose, we take energy, right? We are taking energy from glucose. When glucose is getting metabolized in the muscles, there is accumulation of waste material in the muscle cells. We are not letting the cells to recover because we are constantly keeping it in the active state. We are not letting it rest. So when we are keeping it at work for in extended period of times, the accumulation of waste will lead to the improper functioning of the cells. And at the end of the day, to the muscles. So when the waste starts getting accumulated, our cells are not able to function properly, and hence our muscle feel tired and fatigued. So we say muscle fatigue. So this is what muscle fatigue is. Fatigue in general, we use very generically that we are fatigued, as I mentioned earlier, that people say that they are feeling fatigued or they are tired. But you mentioned that it is not associated with muscles. So I just wanted to clarify that it is not, it's not completely out from the muscle. We still have muscle fatigue. Okay. So in the diagram, you can clearly see what sarcopenia is. This is the healthy tissue. Can you see this? I hope that this diagram is clear, right? So this is the healthy muscle cell, muscle mass. You can clearly see that this is the extent our muscle is distributed, and these are the bones. This is the top section. For example, if I have this arm, if I cut it like this, and if I'm seeing it from the top view, right? So this is what my orientation will look like. This will be my, if you remember our integumentary system had 3 layers. Below that, we had adipose tissues, blood vessels, and then we had muscles, right? Now everything you guys should try to connect. So you can clearly see that this is the outer layer, our integumentary system part. This is the adipose tissue, and beneath the adipose tissue, we have a muscle, and in between we have bone. So if this muscle, the mass of the muscle, and at the end of the day, the force of the muscle decreases in sarcopenia, you can see here, right? So the muscle which was like this, for example, if it is your bicep muscle or tricep muscle, it was supposed to be like this, but due to sarcopenia and loss in the muscle mass, it is now looking like this. Can anyone give me one reason why sarcopenia can happen? I don't want any scientific proof for it, but again, it's coming back to a very, very basic observation which we can make on macro system. Why a muscle, I'm giving you like some hints now. Why a muscle degenerates? Autoimmune, again, sorry, Koshi, right? Huh. Koshi, it's not wrong, but see, fundamentals. Always stick with fundamentals. Autoimmune is a disease which there are diseases which leads to muscle mass, loss of muscle mass. Autoimmune disease, I agree with it. But again, those are exceptional conditions. What I asked very basic, which you have observed in throughout the evolution, that why certain muscles have degenerated. Increased age or older people. Old age is reason that muscle degenerates, but even in young ones, we are able to see degeneration of muscles, right? Disease are often there in which the muscles have been degenerated. I'll give you another hint. If there is a accident, for example, someone has injury in their leg, correct? Kajal mentioning atrophy, right? Yes, polio disease we can see. A very basic thing which I wanted to you guys to think in the direction was this. Muscle follow very basic trend. If you are not using it, they will degenerate. I'm not saying it is sarcopenia. No, sarcopenia is a disease condition. But in general, our body has been customized in such a way that if you're not using something, our body accepts it that, oh, this part is not useful anymore. So why we should supply our resources towards it, right? So now from now on, we'll reduce our resource supplementation towards it. And at the end of the day, that part starts to shrink or atrophy, right? So this thing is very common in muscles also. So everyone, whoever tried going to gym, tried to build their muscles, right? At the end of the day, if you're not able to build up muscles, or say you have built up muscles, but gradually and later they were not that prominent. You can see this in bulkier guys, right? That those who used to go to the gym, but now they are not allowed, they are not able to go to the gym. So their muscles have lost the structure which they had earlier. So they just lose muscles now they have. Why? Because muscle is still there. Earlier also it was muscle, later also it is muscle. But it is reduced. Why? Because they are not exercising it. They are not using it. Right? So the moment in our body you stop using some system, our body adapts to it. And if it is not a survival system, right? Because then you will feel sick and your body will try to jog you up that, no, this is an important system. You are messing around it. Don't mess around it. Take care, right? But if we look at a very small things, like there are many people, if you know, there's a vestigial muscle here. Many people are able to move their ear without like this, right? Like just like cows can do it. Those are vestigial muscles. So in cow, those muscles are still active. That is why they are able to do that. For us, we didn't need it. We are okay without it. That particular muscle doesn't uh, like have any purpose in our survival or in our day-to-day life. Hence, we don't have that muscle, but cows have it. And if you look at other bovine animals, their whole skin, whenever a bee or a mosquito is around them or on them, you can see that they're able to shake their skin, right? You guys are following me, no? Give a thumbs up. I'm saying, have you seen cows and buffaloes when they're chilling and they are sitting, and when any insect or bird or bee comes around, they shake their skin, that particular part of the skin where the bee was sitting or something like this, right? One way is that they use their tail, that I agree. The other is also, you should observe that they shake their localized muscles, right? We don't have that ability. If a mosquito is sitting here, can you only shake this part of your hand? No, right? We cannot, correct? Because we don't have those muscles anymore, because we don't need it. Hence, throughout the evolution, those muscles have been degenerated. You got my point? Great. So we'll move ahead. So our next question is this, I guess everyone should, okay, if you have not followed the lecture, this might be a difficult question, but let's try it. So satellite cells are blank, that blank. We need to fill in the blanks, okay? So what are satellite cells? So satellite cells are stem cells that differentiate to myocytes, or satellite cells are fully differentiated cells that are derived from myocytes, or satellite cells are fully differentiated cells that differentiate to myocyte, or last, satellite cells are stem cells that are derived from myocytes. So I'll give you 1 minute for this, and also I want you guys to focus on other options only, that what those options are trying to say and how they are different from each other. Your 1 minute starts now. I don't have any answer for this question. Wild guesses, guys, wild guesses. Don't worry about it, whether it's right or wrong. Be free. Satellite cells. Now try to relate if you have little bit of idea also what satellite cells are. Then we are saying stem cells. So we are talking about cells. So something has to do with maybe the stages of muscle formation, something like this. Try to approach this question in this manner. Okay, I'll give you 10 more seconds and then we'll see the right answer. We have few answers. Yeah, but I want everyone to at least attempt. It's okay if it is wrong. What will happen? Nothing, right? Just type your guess. A, B, C, D, what? Okay, so without spending more time on this, the correct answer is satellite cells are stem cells that are differentiated to myocytes. Okay. This would have been an easier question if you guys have went through the lecture, because sir has taken, I guess, quite some time in explaining these things. So again, with the fear of repeating myself again and again, do go through the weekly lectures, then attend like open discussions, be active in discussions, then only you guys will be able to understand. So satellite cells are basically stem cells which are around your muscles and different places also. They transport here and there, which at the end of the day, they give rise to myocytes. If you guys are not aware of differentiation or stem cells and those things, you let me know maybe in the next session, because it's a constant theme. Last lecture also we tried to understand some totipotency, pluripotency and differentiation. So if majority of the guys are not familiar with it, I will take 10 to 15 minutes maybe in the next class to let you guys be familiarized with these topics. Okay, but for that you guys have to tell me that you want it or not. Okay. So I don't think so that I have to discuss much into it, because if you go through, and now you have to go through the lecture, that is why I'm not discussing it with you guys. We'll discuss this in the next class if you guys have any doubt in satellite cells. Okay, done. So we'll move towards our next question. What it says, which of the following muscle type does not contain gap junctions? Is it smooth muscle, cardiac muscle, skeletal muscle, or none of the above? Even if you guys don't know the exact answer, and if you are listening to this current discussion actively, you might land up with a good guess. So your 1 minute starts now. Okay, I have few answers coming in. Which of the following muscle type does not contain gap junctions? Twenty seconds to go. Still all I guess most of you have answered it now. Most of you have answered it. Most of you have answered it. Yeah. So the right answer is skeletal muscles, right? So we don't have any gap junctions in skeletal muscles. You can see in the live imaging, maybe not live imaging, but the stained image of a skeletal muscle. So what are gap junctions? Anyone want to tell me? Anyone want to take a wild guess? And I can see that people were confused between A and C, smooth and skeletal muscles. Okay. It's a fair confusion. But what are gap junctions? Anyone has any idea? But have you heard about this term, gap junction? If you have heard about this term, then just give a thumbs up. No one has heard about gap junction. Okay. Okay, Narendra has heard about. Narendra, if you're comfortable, would you like to tell us what gap junctions are? You can either unmute or you can type in the chat box, whatever is comfortable with you. And if you're typing, you need to tell me that you are typing. Okay, no problem. Don't be sorry, please. Nothing to be sorry about here. Permit direct cell transfer of ions and small molecules. Gap junctions. In a broader way, we can say that. Okay. So junctions. Okay, it's a big topic for me to cover, but okay, I'll try. See, we have cells. Now you guys have to be active in this because I'm trying to simplify to the maths here. Okay. So you guys have cells. Cells are in contact with each other. They come together, right? Then they make tissues, then they make organs, then organs make organ system, the basic hierarchy. So when cells are coming together, right? They are in contact with each other like this. Just take example like this, right? So this is A cell, this is B cell. So if A cell and B cell are together, so what do you guys think? There has to be any communication between them or there should be no communication between them? Type in the chat box. My question is cell A and cell B. They are coming together to make a tissue or an organ because this is individual cell, this is individual cell. Now we need to make this complex organism. For us to be here like this, small cells have came together, right? So when they're coming together, this A cell and B cell, should they be in communication with each other? Yes or no? Type in the chat box. I'm asking, I'll repeat my question and I want your responses. Cell A and cell B are coming together to make a organ or a tissue, right? So if they're coming like this is cell membrane. Everyone knows the structure of cell, right? Give a thumbs up if every one of you knows the structure of cell, the basic structure of cell. Any cell, prokaryotic, eukaryotic, irrespective of that. Give a thumbs up if you are aware of it. Without your response, I'm not able to discuss and tell you the extent of things. I need feedback. Otherwise, I'll just pass through this question. I don't mind. Because I don't know how much you guys don't know because you haven't heard about gap junctions. I'm trying to explain gap junctions to you. So I want to know how many of you know the cell structure. And I don't want detailed explanation of it. I just want to know, have you like not seen, but at least aware of basic cell, that there's a membrane, there's a nucleus, there are organelles, right? These basic things. So have you heard about it? Have you seen it? Have you read about it? Okay, continuing. So this A cell and B cell, they need, when they're coming together to make a complex organism, if they are not in contact with each other, so this cell A will do whatever it wants to do, the cell B will do whatever it wants to do. But I'm speaking, right? So my lips are moving in such a way that they're in the coordinated manner. If I'm not moving in a coordinated manner, it will make no sense to you guys, correct? Which means there has to be a coordination. That coordination is coming from muscles. Muscles are made up of cells coming together. At the end of the day, we want cells to be in contact with each other, correct? But cell A and cell B, for example, cell A received some different signal. For example, cell A is from my left hand, cell B is from my right hand. My my left hand has different stimulus right now, like it wants to be in the fist form, but my right hand wants to be in the open palm form. So the stimulus is different for both these organs, right? Both these, my hands, which means the cells which are present here and the cells which are present here are facing different situations, correct? So can we bring down this macroscopic observation to microscopic observations on a cell? So this is A cell, this is B cell. Chances are this A cell is getting some different stimulus, this B cell is getting some different stimulus. So if they are completely merged like this, then there's no distinguishment between these two cells, right? So to make sure that they have limited communication, because for coordination, we need communication. But a lot of communication will disturb the integrity of the cell. So to maintain limited communication, we have certain gap junctions in cell, which will allow restricted communication between adjacent cells for better communication and organization and coordination, right? So that is the function of gap junction. There are different junctions also, but this is one of the junction. Okay. Were you guys able to follow till here what I tried to explain for gap junctions? If yes, give a thumbs up. Okay, great. So we'll move to the next question. Our next question is, pacemaker cells are present in. This is one of my favorite topics. Okay, have all of you might have heard about pacemaker, right? So what we'll answer this question and then we'll get into it. So pacemaker cells are present in kidney, cardiac system, skeletal muscle, none of the above. Everyone should answer this correctly. Your 1 minute, I should not give 1 minute, but okay. Okay, I'm receiving answers. Sneh, do you have a question?
Yes, ma'am, but it is kind of out of track. So can I ask,
Yeah, go ahead.
ma'am, how the study of physiology is different from ours? Like, physiology is common course in medical science, right? Those who are studying MBBS, they are studying physiology, right? So how it is different, like approach of studying physiology, how it is different from ours to them? Like, how do they study differently from us, or how we differently study from them? Can you elaborate this if you have an idea?
So the question is that we both, like the non-medical students and medical students, they both study physiology. So how it is
Yes,
different, right? This is
yes,
what you
yes,
mean to ask.
yes,
Can
yes.
we take this question in the end when we'll
Okay,
discuss?
okay. It's fine. It's
Okay.
fine.
Yeah, no problem. We have studied it in ECG. What is ECG, Kajal? Like, I'm not aware of this. If, like, I know what ECG is, but do you mean the same ECG which I also mean? So I want to know that. Yeah, so just text me. Okay. So obviously pacemaker cells are present in cardiac system. You might have heard many people that they have a pacemaker installed, right? So what pacemaker does, it helps in regulating the or normalizing the heartbeat for the patients who are suffering from irregular heartbeats, right? Okay, yeah, okay, yeah, Kajal. So it looks something like this, that we have a thread, we have sensors which via operation we put inside the area which was dysregulating or which was not able to contract, or like on timely contractions were not there. And then we have a what to say, another sensor which will transduce the signal whenever the internal sensor will detect some abnormalities. You guys will understand this, like I have this very small video of how a pacemaker works. So I hope this will be exciting for you guys as well. Just let me know if you guys are able to see my screen. Not till now, I'll ask you. Okay, so can you guys see my browser window? Can you guys see my browser window, a video? Okay, now you guys have to tell me also whether you guys are able to hear the audio. I'm gonna play it, then stop it, and I'm gonna ask that are you guys able to hear the audio as well, okay?
Electrical activity within the heart determines heart.
So were you guys able to hear that? Okay, done, great. So I'll start the video and then we'll see where it land us, okay?
Electrical activity within the heart determines heart rate. If this electrical activity is delayed, a slow heart rhythm may occur. People who have slow heart rhythms may feel tired, dizzy, lightheaded, or even pass out. A pacemaker is a small medical device that is placed under the skin, below the collarbone, and is used to treat symptoms related to slow heart rate. A pacemaker is composed of two parts, a pulse generator and a wire or lead. The lead is passed through a vein and makes contact with the heart muscle. The pulse generator constantly monitors the heart rate. When it detects that a heart rate falls below a certain level, it sends an impulse down the lead to the heart, which causes it to beat. A small surgery is required to implant a pacemaker, which takes about an hour. Patients are awake during the procedure and are given a mild sedative. The area under the collarbone is frozen and a small incision is made. The lead is then passed through the vein to the right side of the heart using X-ray guidance. The pulse generator is then connected to the lead, and the incision is closed. Patients will usually go home the next day. Pacemaker surgery is safe. The most common risks are bruising, bleeding, or pain around the incision. Other risks include infection, or a pacemaker lead moving, requiring another procedure to reposition it. After your operation, you will be given instructions on how to care for your incision and will be given an appointment to come back to.
Okay, so you guys are able to see my presentation now. Okay, cool. So you guys were able to follow what a pacemaker is. It is interesting, right? And just imagine that this was some like quite long ago. Now we have literal patch-like pacemakers. They are not called pacemakers, but they do the similar function of a pacemaker. The basic function of pacemaker is to regulate the like sense when the heartbeat. Now what heartbeat is? Heartbeat at the end of the day is a relaxation and contraction of a muscle, which we are studying in this thing, right? So relaxation and contraction of a muscle when it is not regulated or it is not what it is supposed to be. So it is sensing that, sending electricity. Imagine we started our discussion with electricity itself, correct? Sending electricity to assure that our muscles are relaxing and contracting at the rate which we want it to be, or the like physiological condition our system is attuned for, correct? So pacemaker cells are present in cardiac system, as we can see here. This is somewhat trivial system. The other modernized and minimalist systems are also there through which you can regulate your heartbeat from outside itself, and you don't need incisions or surgeries. It is a surface level approach via sensors and light and electricity and LEDs and all those. There are multiple things which are going on right now. Okay, so we'll go to our next question. Our next question says, which of the following is not a striated muscle type? We are discussing this since the beginning, so all of you should answer this question. You have 1 minute. Striated muscle type, not a striated muscle type. I can see many people are confused between smooth muscle properties and skeletal muscle properties. We will I guess I have put that image, which was also in the earlier slide. It might help you in understanding, clearing the concepts of it. Yeah, so a not striated muscle is a skeletal muscle. If you guys have confusion, go back to the lecture slides first. These confusion won't be here anymore if you would have gone through the lectures, but it's okay, no problem. Go through the lectures, your basic doubts will be cleared, I'm sure. For those who are answering correctly, kudos to you guys. For those who are attempting, a big kudos to you guys. For you guys who are not able to get the right answer, don't worry. Okay, by the time the course will end, you will definitely be having some positive knowledge and information about physiology. So this is a very brief table about skeletal muscle, smooth muscle, and cardiac muscle. Skeletal, smooth, cardiac, as during the beginning of the lecture, Mansi very well summarized this week's lecture. So she mentioned this point that this classification of muscle about skeletal, smooth, and cardiac is dependent upon the force they generate, right? So if we change our frame of reference for classifying something, same thing can be named in different manners, right? For example, if my classification is how many students are there in my current session, right? Then there is only one group. I'll say this number of people are present in my session, correct? Now, if I change the point through which I'm trying to distinguish or classify you all. Now, if I ask the question, how many guys or girls are present in my system, in my session, sorry. The answer will change, yes or no. What I'm asking, I'm saying, if I ask the question that how many people are in my session, I'll get one type of answer. If I ask how many guys and girls are present in my session, will I get the same answer or different answer from the previous question? Type in the chat box. I want you guys to be active. The answer will be different, right? Because I asked earlier how many people are there. I'll say this number of people are present in my session. But now I'm asking how many guys and girls are present in my session. Then I'll say these many number of guys are present, these many number of girls are present. Correct? So same system can be defined in two different setups depending upon how we are asking the question. Correct? Similarly, the skeletal system, the muscles can be differentiated into depending upon what question you are asking. If you are asking, okay, this is A type of muscle, this is B type of muscle, this is C type of muscle. A type of muscle is generating this amount of force, but B type is not producing this amount of force. Hence, we can classify these three muscles in three different groups based upon how much force they are generating. But now I have changed my question. Now I'm asking, under the microscope, how these muscles are looking. How I am able to visualize them, and I'm looking at them, how they are in actuality. Right? So from that point of view, the classification changes to striated and non-striated muscles. Got it? Okay, great. So we have our next question. In skeletal muscle, which of the following prevents interaction between actin and myosin? Chances are that if you guys have not gone through the lecture, you might not know, but don't worry, give it a try. You have 1 minute. In skeletal muscles, which of the following prevents interaction between actin and myosin? Is it myotube, actin, tropomyosin, troponin? I think it's a bit difficult question if you guys have not gone through the lecture. So I guess I won't spend time in this, and I'll just give you the answer, and I'll have a small video for you guys so that you can relate to what these different entities are doing. So I guess we'll quickly go to that only. This will help us in also understanding. And after watching that video, if you guys go back to the lecture, you might understand it in completely different way. I'm gonna share again, so you guys have to respond again. First, I'll share my screen and then I'll share the sound. So you guys have to tell me that. Cool? Okay, so I hope my YouTube is visible to you guys. Thumbs up. Okay, cool. And now you have to tell me whether you are able to hear the audio or not.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin. The The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
Guys are able to see my slides? Yes, no, thumbs up. Okay, cool. So, did you see how we are contracting and relaxing muscles? I know it was a bit complicated if you guys are seeing it for the first time, but the basic idea is actin myosin are contractions, right? In the video, you can clearly see that something is guiding over something, then we are contracting and then relaxing, right? And ATP was associated with it. So that molecular functionality of these proteins, actin and myosin, is leading you to do all the daily tasks, whether it is to get up, get down, pick up something, put down something, anything, whatever you are doing. The moment you are using muscles, the relaxation and contraction is happening, and the relaxation contraction is due to actin and myosin. Okay, cool. So these calcium signaling and all, it's a very deep, or say molecular at molecular level, it's very challenging to understand. So we won't be discussing it that much, but if any one of you interested, you can write it to me. I can send you certain resources if you are interested in it. Okay, so our last question for today, and I guess you'll get the answer even if you don't know the answer about it, but if you have heard the previous video, you'll be able to answer it. So in skeletal muscle, the specific calcium binding site is actin, myosin, tropomyosin, or troponin. I'll give you guys 1 minute for this question. We just saw skeletal muscles and we saw contraction and relaxation. There was some discussion about calcium binding site. That calcium binding site was present on which protein or which entity? I guess I have few answers. Okay. CD. Okay. Okay, yeah, obviously, because we just saw it was the troponin entity, right, which was associated with the calcium binding, leading to the exposure of the myosin head, and then myosin sliding on the actin filaments, leading to the relaxation and contraction, right? These things might overwhelm you, but don't get overwhelmed. Go through the lectures at your own pace, but try to finish the lectures in that week itself. I'm not saying sit tonight and finish this week's lecture. No, go gradually about it. You have Monday, Tuesday, Wednesday, Thursday, Friday, right? So watch one video in 1 day. I'm saying don't even watch 2 videos in 1 day. Okay, it's a half-hour video. So go easy on it. You will be able to go through it with the lecture flow. Okay, don't worry. So now this, okay, this is very interesting. What it is, how many of you can recognize it? Give a, like, raise your hand if you're able, if you know what it is. And I don't want to ask, I'm not going to ask you guys that what it is, exactly what it is, but have you guys seen such images around social media or in conferences or something like that? Yeah, these are the cells stained and visualized under microscope or confocal or different high imaging techniques, right? So all these green and red, which are very dense, can you see the green and red color? Right? This thing, green and red color. These are the actin myosin filaments. Imagine your cell has such a dense connection of actin and myosin, right? And these are all contracting and relaxing at the same time. The moment you are doing giving it some stimulus, doing some work, how beautifully placed they are, right? So even now we are not able to replicate it in the labs, that how intricate this actin myosin communication is. We know something about this communication, but still we don't know 100% of it. So still it's a very active area of research. So with this, I would like to thank you all for your patience and thank you for being active participant in today's session. So now we will just have few discussions. We'll take, I guess Sneh asked the question about the way we are getting physiology, how we perceive physiology in medical and non-medical field. And second, as you guys know, these are certain questions on which I want you guys to think about it. So for this week, it is supramolecular coiling in muscle similar or different as compared to protein folding? This question you will understand if you go through the week lecture. You will understand what supramolecular coiling is. Go through the lecture, understand supramolecular coiling, think about protein folding, and we'll discuss it later. Okay. Coming back to